Hello, hi. Okay, so welcome everyone to the second lecture in our Dwayne L. Ford guest lecture series here at Andrews University, Department of Chemistry and Biochemistry. Welcome all of you. We have some information in the chat. Mute your mics, mute your sound when you come in. If you are a student in the class or seeking co-curricular credit, put your name and present so you could get credit for that. To all of our guest attendees, if you could put your name and affiliation so we could know, get some sense of where people are from, looking in from. At this point, I have the pleasure of introducing the introducer. My colleague, Dr. Novak, he's professor of biochemistry here at Andrews University. And he is the gentleman from Owensville, Missouri. It's your turn, sir. Go ahead and turn on your mic.
There we go. Yes, it's not not the end of the world, but you can see it from there. It's a nice little town in the middle of Missouri. But I get the pleasure of introducing Dr. Wong, and I just am grateful for him being here. Dr. Wong received his BS and MS degrees from the National Taiwan University, and his PhD in 1982 in chemistry with George M. Whitesides, a noted chemist here in the United States, from the Massachusetts Institute of Technology. He then worked at Harvard as a postdoc, again with Dr. Whitesides, for another year, and became a faculty member at Texas A&M University in 1983. Well, he was promoted to a full professor in 1987, which just indicates the rapidity of which indicates the quality of his work. He is currently with the Scripps, he currently holds the Scripps Family Chair Professor in the Department of Chemistry at the Scripps Institute, and holds a joint appointment as a distinguished professor in the Genomic Research Institute at Academia Sinica. Dr. Wong has received numerous awards, the most recent of which are the ACS Arthur C. Cope Medal, the Wolf Prize in Chemistry, and the Robert Robinson Award from the Royal Society of Chemistry in the United Kingdom. He's a member of the Academia Sinica, the American Academy of Arts and Sciences, and U.S. National Academy of Sciences and U.S. National Academy of Inventors. His research interests are in the field of chemical biology and synthetic chemistry, with particular focus on the development of new methods and tools for the synthesis and study of complex carbohydrates and glycoproteins associated with disease progression related to aberrant biological glycosylation. He's published over 750 papers and more than 1,200 patents, and we are welcoming him with pleasure. Dr. Wong, the time is yours.
Okay, thank you very much, David, for your kind introduction, and also thanks to Desmond for arrangement of this lecture. It's really a great pleasure to be with Andrews University by Zoom. So the subject I want to talk about is universal vaccine development through glycoengineering to target SARS-CoV-2 and influenza virus. As you know, the 1918 Spanish flu infected almost 500 million people and killed 50 million. And after 100 years, we see another pandemic caused by SARS-CoV-2, which infected over 200 million people and caused almost 4.5 million deaths. You know that the vaccination has been considered to be the most effective means to control the pandemic. However, we also know that once a virus is in humans to cause the pandemic, vaccination is almost impossible to eradicate the virus, with the exception of smallpox, vaccine, which actually took several hundred years to eradicate the virus. On the other hand, we know that the human viruses are frequently to mutate and heavily glycosylated to escape from immune surveillance. So how to develop a broadly protective vaccine against influenza or the SARS-CoV-2 virus is a very interesting issue, and which is the focus of today's lecture. But before I get into the issue, I want to take this opportunity to describe the impact of glycosylation on the structure and function of cell surface protein. In this case, we'll focus more on human cells and also virus. So here is the human blood cell. You can see that it's completely covered by carbohydrate. The fluffy stuff actually is the carbohydrate, but we don't know what they are doing there. What we know is that the carbohydrate on the surface of red blood cells will determine our blood type. And as you can see from the next slide, that the glycans that determine the blood type are very, very similar, all having the type O core structure there. And you can see that type A and type 2 glycans have a very small difference as indicated by the purple color. And even more mysterious is that type A persons have the antibody against type B glycan and vice versa. And type O persons can give blood to type A or type B person, but not the other way around. And it's even saying that blood type will determine personality. So we actually really don't understand the origin of these antibodies and whether the relationship between blood type and personality is true. So over the years, we have been interested in understanding the role of carbohydrate in biology, particularly on the surface of cells, and then have been actively involved in the development of new tools and methods for the study of these class of molecules. So here is to summarize our recent activities. We have developed a programmable one-part synthesis of oligosaccharide and chemoenzymatic synthesis of glycans and glycoproteins. Design glycosylation probes to identify cancer-specific glycans on the surface, and also develop glycan microarrays for the high throughput analysis of protein-sugar interaction. Now, these methods have been used to study the function of carbohydrate on cell surface and to develop carbohydrate-based medicines. For example, vaccines against cancer and viral infection, homogeneous antibodies with the improved effector function, and also the inhibitors of the enzyme associated with the disease-causing carbohydrate. Now here is the programmable one-part synthesis scheme, which we developed many years ago. I believe this is the first automatic methods for oligosaccharide synthesis. And today we have added more than 50,000 building blocks by machine learning. Everyone has the predicted reactivity. And we also updated the computer program as a guide to select the building blocks for the one-part synthesis. So, for example, if you want to make this oligosaccharide, you simply enter the sequence into the computer, and it will show the building blocks to be used in sequence. I should emphasize in sequence because it's all based on the reactivity. We start with the most reactive one, followed by the second and third and the fourth. So once you add the building block in sequence, then in a few minutes, you will get the product with the protecting group there. And after globally protection and clean up with chromatography, you will get the oligosaccharide for the study. This method can also be used to make oligosaccharides with the differential protecting group, such as the heparin pentasaccharide shown here, to allow us to introduce the sulfate group in a regioselective manner to understand how the sulfate and their position affect the function of the oligosaccharide. So it gives us some flexibility to quickly synthesize different oligosaccharide with the different sulfation patterns. And this kind of programmable one-pass synthesis actually was inspired by the way oligosaccharide is made by enzymes. For example, in the synthesis of global H or SSCF4, both are cancer-specific glycans, the first enzyme product becomes the substrate of the second enzyme. And the product becomes the substrate of the next enzyme. So in principle, you can mix all these enzymes together to get the product in a one-part manner. Even one can include the cofactor regeneration scheme because the sugar nucleotide used in the synthesis is very expensive. So with the introduction of sugar nucleotide regeneration, one can reduce the cost and also eliminate the feedback inhibition caused by the released sugar nucleoside phosphate. So we then look at the impact of glycosylation on glycoprotein structure, and we start with the O glycosylation, and where the N-acetyl glucosamine is attached to the serine or threonine residues. So we use the RNA polymerase II as a model and found that when the threonine residue is glycosylated with N-acetyl glucosamine in the beta linkage, it induced the local structure from a random coil to a type 2 beta-10 structure, and that serve as a nucleus to facilitate the folding of ribonucleus, RNA polymerase. We also look at the glycosylation of EGF2, a methyltransferase involved in the methylation of histone 3, lysine 27, to inhibit tumor suppression. So there are about 6 glycosylation sites. And these 4 actually are important for the stabilization of the protein. And the last one at the position serine 729 is essential for not only the folding, but also the enzyme activity. And then this is very, very interesting because there are many proteins which have all glycosylation there, and we don't quite understand what they are doing. In some cases, you will see that all glycosylation is competing with the phosphorylation in the signaling transduction process. Now, then we look at the N-glycosylation and found that the first sugar, again, N-acetyl glucosamine, is the most important in terms of stabilizing the protein. It speeds up the folding by about 0.8 kilocalorie and stabilize the folded structure by about 2 kilocalorie. Almost 2/3 of the total energy involved in the stabilization of this model protein, human CD280, which has only one N-glycosylation site, and there's no protein or disulfide bond to interfere with the folding pathway. So this is a good model for the study. And this core trisaccharide is highly conserved in eukaryotic species, ranging from yeast to human, and they are involved in the folding of the protein. And the rest of sugars actually are not associated with the folding, but they could be involved in recognition, and so is the core glucose there. So we want to know what is the origin of the 2 kilocalorie contributed by the N-acetylglucosamine. And we use NMR to look at these interaction in a dynamic manner. And we found that the N-acetylglucosamine actually has a hydrophobic interaction with the nearby phenylalanine group. And this hydrophobic interaction cause the change of orientation of the phenyl, the benzene ring here. And as a consequence, it triggers the following hydrophobic interaction in the beta sheet. So it's more like almost like a domino effect. And this interaction, the sugar phenylalanine hydrophobic interaction is about 1 kilocalorie. And the following cascade is another kilocalorie. So this is the origin of the 2 kilocalorie contributed by the glycosylation of this asparagine with the N-acetylglucosamine. There are about 20,000 N-glycans commonly found on human glycoproteins, shown here, including bi, tri, and tetra-antennary N-glycans. And these glycans are very similar and very complex and difficult to obtain or to synthesize. So we have developed a modular approach to the synthesis of these complex N-glycans, and we divide that into these 3 modules with the mannose at the reducing end, and we create oligosaccharide monosyl chloride as a building blocks for connection to the core mannose here. So with the 3 kind of modules available, one can assemble all kinds of N-glycans here. And this module can be synthesized chemoenzymatically. And usually what we do is we introduce cyanotic acid or glucose at the end, and then do the complete deprotection, and then we acetylate the glycan again and convert them into oligosaccharide chloride for coupling. So these are very convenient methods which can be used to quickly assemble different kind of N-glycans. And with these N-glycans available, one can make sugar arrays. And here is another method we introduced for the synthesis of cyanotic acid containing oligosaccharides for sugar array preparation. So we start with this kind of phosphate with these 2 groups tie up together, and that will enhance the alpha glycosylation of the cyanotic acid. And either you use the orthogonal 1-part synthesis. Here we use phosphate and other sugar could be different group, or we create a disaccharide first, because cyanotic acid usually is the least reactive glycosylation unit. So we create a disaccharide first and use that as a building block for the 1-part programmable 1-part synthesis. And in this way, the reactivity now is determined by the second sugar instead of cyanotic acid. So with these sugars available, we can assemble the glycan array. And here is to show how to use the array for the study of hemagglutinin binding to understand how the hemagglutinin from different influenza virus recognize its receptor, that is thionyl glycans. We can also create a mixed array using aluminum oxide coated glass slide because the surface is more homogeneous, so you can create, in this case, every spot has 2 sugars, and then you can look at heterodigam binding, and here is the example about PG9. It's an antibody isolated from HIV positive patient who never get into AIDS. And so we thought that the reason is that because the patient had the antibody to control the replication of the virus. So if we understand the epitope of this kind of antibodies, we go broadly neutralizing antibody. We may be able to use the epitopes to design the vaccine and to elucidate PG9-like antibody to neutralize HIV. So here is the hybrid type, the unusual hybrid type glycan identified from the array, which is recognized by a PG9 antibody isolated from HIV patient. So with this background, I want to get back to the universal vaccine design against influenza and SARS-CoV-2. And these 2 viruses are RNA virus, which is easy to mutate. And the surface of the virus, either hemagglutinin or spike protein, is highly glycosylated to facilitate infection and also to evade immunity. So our proposition is that if we remove the sugars, particularly those which are unnecessary or which will not affect protein structure, then we can expose the peptide epitopes, particularly the conserved epitope, to the immune system and to increase the immunogenicity. So that's a very simple idea. And then we look at the trend of mutations. In this case, we look at COVID-19 virus, and you see that the key mutation is this one, D614G. Aspartic acid at position 614 of the S protein is mutated to glycine. And this mutation will increase the transmission rate by about 10-fold. And further mutation going to alpha, or the most predominant variant today, the Delta, they are all from this one. And as the virus continue to mutate and transmit, the current vaccines are based on recent observation are fading. As you can see that this is a real world situation, it's not the clinical trial data. So for example, the Pfizer and Moderna vaccine now have a decrease by 6-fold and 2-fold against Delta variant. And the Johnson & Johnson and AstraZeneca even less effective. And Novavax is the use of S protein as a vaccine, but we don't have the data yet. The phase 3 trial looks very promising, and we are waiting for the approval of this protein-based vaccine. But in any event, you see that when the virus continues to mutate, the efficacy of vaccine is going down, even though they are highly effective against severe disease, but it's impossible to completely protect against infections. So here is to show the glycosylation of the spike protein. And some representative mutants and the mutation site. There are about 2.5 million S protein sequence reported by GISAID today, although most of them are not significant because the mutation rate is very low. And more than 95% of the confirmed cases today are infected by the 4 variants of concern shown here, including the Alpha, originated from UK, the Delta, originally from India, Gamma, and the Beta strain. And of these 4 variants of concern, the Delta variant is the most dominant. It accounts for more than 90% of the total confirmed case we know today. And from the glycosylation pattern, you can also see that most of the surface area which are conserved actually are glycosylated. And the light color means the mutation is has a low frequency, and the heavy color means the mutation rate is higher. So this gives you an idea about where are the conserved epitopes which can be attacked by either antibody or T-cell response. And here is to understand how the glycosylation on the S protein affect its binding to the receptor. We know the receptor is angiotensin converting enzyme 2, so we express the receptor on different human cells. These are 293 T cells, and here is a brittle E6 cell, that's a kidney cell, and these 3 are the cell lines from airway, and we construct a pseudovirus to mimic the real virus, and with the spike protein on the surface, and then we delete each of the glycoside. I should mention that there are the S protein is a trimer. Each monomer has a 22 N glycoside and 2 O glycoside. So we delete each glycoside at a time, and then see how the deletion of that glycoside affect the binding. And as you can see that almost every site has some impact on the binding, and particularly the sites in the RBD region and the site at the 801 and 1194 are very significant in terms of effect on the binding. And we also found that if the S protein is expressed from human lung cells, and which are the primary site of infection. We know the virus infect human through the epithelial cells in the airway. So if the virus, if the S protein is expressed from the non-epithelial cells, it contains more cyadylated complex type N glycans, which actually are more effective than the other type of glycoforms. So this is a very interesting finding. That is, S protein expression is cell specific. It depends on what kind of cells you use to express the S protein. The glycoforms are different. And the glycoforms from the airway epithelial cells is most effective because it contains more cyadylated complex N glycans there. So here is a comparison between the S protein from non-epithelial cells and from HEC293 T cell. This is a common cell line used for S protein expression by many other laboratories. And you can see that these two actually are different. I should point out that you may pay attention to the yellow and the green color. So these two are different. And the one from HEC293 T cell has more hybrid type. and more mannose type of structure glycan there. And here is the individual site. You can see that, for example, N17 has mainly the, uh, the complex type. So between these two cell lines, but they are actually different in some area. Like, for example, this position is completely different. So this difference in glycosylation, of course, will affect the infectivity of the virus. And then we take about more than 1 million sequence of S protein reported by GISID, and then look at the conserved epitope on the surface and to see if they are still available for immune response. And we found that the most conserved areas are in the RBD, receptor binding domain, and also the H2 domain in the C terminal. But almost 50% of these conserved epitopes are shielded by glycans. So in other words, even though the RBD and HR2 are the good epitopes for vaccine design, they are covered by sugars. So basically the immune system cannot see them. But of course, it depends on the processing of the vaccine in dendritic cells. Sometimes if dendritic cell has enough glycosylates to remove the sugars before presentation to T cell, then that will induce a stronger immune response. But in general, glycosylation on the cell surface is a problem. So what we do is we prepare the fully glycosylated S protein, and this is equivalent to the vaccines we use today, either DNA vaccine or RNA vaccine or the vaccine manufactured in vitro. And then we use endoglycosylates to remove the sugar and keep the N-acetyl glucosamine at every N-glycoside because that is required for the intake of protein structure. And we use these two as an immunogen for immunization, as shown in the scheme here. And after that, we collect the blood and then measure the IgG titer, the anti-S protein activity, and the neutralization activity. The neutralization activity is done by pseudovirus in most cases. That is, we use a pseudovirus to infect human cells which express SE2, and then we add the antibody to that and to see how much it can be protected. And we also do single B cell analysis and found that the B cells from monoglycosylated spike protein has more variety, particularly in the light chain. You see that there are more B cells in the light chain of the antibody. And the neutralization activity and the anti-IgG titer are better from the monosaccharide vaccine. And we also look at the B cell and CD8 T cell response after immunization with the fully glycosylated and monoglycosylated S protein. So as mentioned before, the anti-S IgG titer is higher and neutralization is better for the monoglycosylated spike vaccine. And then we look at the Th1 and Th2 ratio. It's an indication of how good the CD4 and CD8 response is balanced. And again, we see more Th1 than Th2 as measured by the IFN gamma secretion. And And this IL-4 is an indicator of a Th2 expression. And then we also look at the secreted cytokine. So granzyme B is a measure of CD8 T cell response. Uh, and it also shows more anti-S B cells from the monoglycosylated S protein. So all these indicate that the monoglycosylated spike uh, vaccine will induce more B cells and stronger CD8 T cell response. And here is the study of the binding, neutralization, and potency against the virus and variants, particularly the 4 variants of concern, as I mentioned before. So you can see that the monoglycosylated vaccine is always better by 2 to 4-fold than the fully glycosylated one. And here is the challenge study. You see, it's also better. And in the case against the alpha strain and the delta strain, against the alpha strain, it's almost fully protected. And against the delta strain, it's almost 90% there with the lethal dose of the virus. And the fully glycosylated one is not as good as the monoglycosylated. So again, it shows that the removal of the glycans from the S protein will create a much better vaccines, which are more broadly protective against SARS-CoV-2 and the variants of concern. So then we use a single B cell technology to isolate antibody. And here is one example we call M31A7. This antibody cannot be found with the immunization of fully glycosylated S protein, probably because of the shielding of the glycan at the N165 position, but it's induced in the monoglycosylated S protein immunization. And the reason this antibody can bind to the RBD conserved epitope so well is because it approached from the position opposite to the glycan here, and then slowly grab the epitope here. And you can see that the dissociation constant is in the picomolar range. So this antibody is targeting the highly conserved epitope in the RBD domain and is able to neutralize almost every variance of concern we know today in the picomolar range. So this is a very exciting result and also support that the removal of glycans on the surface of immunogen is a very effective way of designing a broadly protective vaccine here. So here is to summarize the immune response process. Once we get the glycoprotein vaccine, in this case is the monoglycosylated one, you'll be taken up by the dendritic cells, which then process the glycoprotein by protease and glycosylase cleavage, and then present that to T cell, to MSC complex. So if the glycopeptide are mixed with MSC1 and presented to killer T cell, this is we call CD8 T cell response, because the killer T cell will kill the virus directly. If the presentation is to helper T cell or CD4 T cells, you will tell the the B cell to make antibodies against the virus. Now, in some cases, we also use adjuvant to further activate the whole process. But in our study, we just use aluminum oxide because we just want to understand the basic of the immune response here. But if necessary, we can use a better adjuvant. And this is the glycolipid adjuvant design in our laboratory to enhance the Th1, Th2 ratio and also to stimulate the class switch from IgM to IgG. But we didn't use this one here in the study. So this is the summary of the development of a broadly protective vaccine against SARS-CoV-2 and variants. And the strategy actually was first used for the development of universal vaccine against influenza. Again, the spike protein is hemagglutinin in the case of influenza virus. And hemagglutinin is also a trimer. And each monomer has a 6N glycoside. It's less complicated than the SARS-CoV-2. And we found that the asparagine 27 is essential for folding of hemagglutinin, and position 144 is essential for binding specificity. That is, in the case of human virus, it requires this glycosylation there to ensure that the hemagglutinin recognizes 2,6 linked sialocyte. We also found that the region covered by sugars are highly conserved. And that's also similar to what we found in the case of spike protein. So again, our initial thought is to remove the unnecessary sugar and keep the gluconate there to make sure that hemagglutinin is still a trimer. And then here is just to show the linkage. Human influenza virus recognize the 2,6 link sialyl oligosaccharide, and the 2,3 link is recognized by avian virus. So we express the H5N1 hemagglutinin in GNT1 deficient 293 T cells, and then treat that with the NOH to create all the N-glycan down to N-acetylglucosamine only. And this monoglycosylated HA is a very effective vaccine again. So you can see that after immunization, the anti-serum or anti-antibody has better neutralization activity. And in the challenge study, it's also much better than the fully glycosylated hemagglutinin as a vaccine. And most recently, we create a chimeric one. We use a consensus H5 as the head, and then consensus H1 as the stem. And this chimeric hemagglutinin is expressed, and then the sugars are trimmed down to the monosaccharide state, as shown here. What I mean about consensus is that we do sequence alignment and starting with in the case of H1, we start with the sequence from Spanish flu in 1919 18 up to now, and then come up with a consensus sequence, and then we take the stem region of the H1 and the head region of the H5 to construct the chimeric. And this vaccination of this monoglycosylated chimeric HA. In a Chinese study, we show that it's very effective against H1N1 and H5N1 variants and subtypes. And in the neutralization study, it's even broader. We look at H1, H3, H7, H5, and H9, and all are very effective in terms of neutralization activity. And here is the challenge study. Indicated in red is the monoglycosilicon chimeric one. It's highly protective, except the Solomon strain, which actually is not circulated now. So we don't quite understand why it has this effect against the Solomon strain. Now here is the illustration of antibody improvement. We know the antibody FI6 from Cosell is a universal antibody against almost all influenza type A viruses. So we take that antibody and again engineer the FC glycan there. We first create that to the monoglycosylated antibody and then do transglycosylation using glycan oxazoline as a substrate to make different kind of glycoform, homogeneous antibody glycoform, and to see how they affect the effector function. We know antibody has two functions. One is a binding to the target, and the other is to recruit the immune cells to the target, to kill the target. And that requires the glycosylation in the FC region in order to recruit the immune cells. So the type of sugar here will affect its affinity to the receptor on immune cells. And we found that this biantennary glycan with 2,6-iodidated N-glycan here is the best one for ADCC, the antibody-dependent cytotoxicity. And you can see that compared to the FI6 antibody, the cytotoxicity and ADCC is much better, and even in a challenge study, the homogeneous antibody is much better than the antibody originally identified by Cosell. So, as I mentioned, antibody has two functions. One is to capture the target. And once the target is captured, you have to kill it. And the way the target is killed is through the recruitment of the immune cells, for example, NK cells or macrophage, to the site of infection or to cancer cells. And that interaction requires the proper glycosylation here. So the FC region of the antibody can interact with the FC receptor on the immune cells. And if you want to have better ADCC, you need to have the glycan as shown here. And if you want to have a better CDC or vaccinal effect, you need to have different glycan here. So I think with glycoengineering methodology established, one can optimize any therapeutic antibodies for different purposes. And we have the structure of SCT attached to the FC region, and we found that the glycan is more attached to the protein. In that way, it has better interaction with the receptor. So here, this is all what I want to share with you. And my conclusion is that the removal of shielded glycans from viral surface glycoprotein to better expose the highly conserved sequence to the immune system is an effective and very simple strategy for universal vaccine design. And thank you very much for your listening.
Thank you. Thank you very much. Okay, so we will see if we have questions from our audience. You guys could put your questions either in the chat or you could unmute yourself and ask your question. Identify yourself, you know, your name and so on when you ask your question. So you can go ahead. I see one hand here from Michigan State. Yes.
Hi. Yeah. Hi.
Yes.
This is Shafiq Huang from Michigan State University. Thank you, Dr. Wang, for sharing this exciting results. I really enjoyed it. So a quick question. So you're thinking the monoglycosylated one, it just exposed the protein epitope that's been covered. But could that be also create a new epitope with the monosaccharide gluconate attached to it? It's a novel epitope that's been recognized better by the immune system.
Yes, that actually what we worry about in the beginning, and we get into the detailed analysis of the T cell and B cell response, and we didn't see the so-called antibody-dependent enhancement there. So most of the antibody or T cell induced are specific for the target, and you just see the increase in the level of antibody or T cells, but you don't see the antibody or T cell that target the unnecessary part. They don't target sugar, they target protein. Protein are foreign, so it's okay to target the foreign proteins, even though the, the intact S protein or hemagglutinin are fully glycosylated. And some antibody may not be necessary. But as you know, sugars are more flexible. And once there's a contact of the antibody and proteins, then thermodynamically you will push the sugar away and to make protein-protein interaction stronger. So I think all the antibody generated have some kind of affinity to the peptide part of the target protein. And that make the immune response stronger. And that's how we explain that. But in the real case, we use a fully glycosylated S protein or hemagglutinin for the assay. And you do see that most antibody will bind to the protein with fully glycosylated. And to explain that, I think is to do with the flexibility of the sugar.
I see. With your glycoprotein, even after deglycosylation, you still have some O-glycan on it, right, for the spike protein.
Oh yeah. In the case of hemagglutinin, there's no O-glycans. But in the case of spike protein, there are at least two there, and we didn't touch them because the O-glycans are shorter, so we don't think they will have much interference with the access of the antibody.
Great, thank you.
Okay. Anybody else has questions? Again, if you want, you can unmute your mic, your sound, and ask your question.
Hello.
Okay, go ahead.
Hello, Dr. Wang. Thanks for the talking. I'm from Shafiq Huang's lab, and I have a few questions. The first one being maybe related to Dr. Huang's question is, so why do you think in the fully glycosylated case, those epitopes shielded by the glycan are not recognized by the BCR? because in your case, you show that the antibodies generated from monoglycosylated epitope can recognize those shielded epitopes even on the glycosylated protein, right? You say they can push away the glycan maybe somehow and recognize those parts. But in the glycosylated vaccine, those parts are seemed not recognized by the BCR, those not targeted by the human antibodies. So why do you think there's a difference in terms of BCR recognition and antibody recognition?
Yeah, I think it's to do with the, the mechanism of antigen presentation. And I show the scheme of immune response pathway. So when a fully glycosylated S protein is taken up by dendritic cells, for example, the dendritic cell has the protease and glycosylates to cleave the glycoprotein, right, into glycopeptide, and then package it with the MSC for presentation to T cell. So it depends on the individuals, and sometimes we don't have enough glycosylates to process the sugar. So if the presentation of peptide is still has many sugars attached to, then the immune system cannot see the peptide epitope clearly. So the response will be weaker. And that's how we explain. And that actually is the case in vitro. You can take a fully glycosylated, monoglycosylated as basically affinity ligand, and then see how they interact with the antibody generated. And you will see there's a big difference between the monoglycosylated and fully glycosylated, as we see in the single B cell analysis. Very different compositions and B cells there. Yeah.
Okay.
So,
I have another question. Sorry.
okay.
So the second question is that you showed that those sequences shielded by the glycans are highly conserved.
Oh yeah, yeah.
Is that a result of lacking selection? Basically, since our human antibody did not recognize those epitopes very well, so they are not under pressure. If we choose to expose those epitopes, the other virus going to evolve and mutate away.
Yes, yes. I think in the case of influenza, you start to see that the glycoside change, and you see more and more glycosylation site are coming. But in the case of S protein, the spike protein, we have not seen any change of the glycoside. These glycosides are all highly conserved, and the sequence around the glycosylation site are also highly conserved. We know sugars are from human, not from the virus. So sugars are T cell independent. They are immune silent, and we have no idea why the highly conserved epitope is shielded by glycans. Maybe they are not under the pressure of antibody or immune response, so they don't have to mutate that areas. But I think when time goes, it could change because in the case of influenza, you do see that. Yeah. But even that, when you remove the sugars, you expose more more peptide epitopes, and that will induce more immune response, and that's for sure.
So the focus, so are you okay?
Yeah, that answered my question. Thank
Okay,
you.
good. So the focus of your talk was directed at the glycoengineering of for viruses. Is this something that's more broadly applicable So for example, my understanding is that there's an overabundance of sialic acids on cancer cells compared to normal cells. Does your technology of glycoengineering is something that could be applied to that problem as well, that disease as well?
I think so, but I think we have to get into that to make sure it's generally applicable. For example, there are many other human viruses like dengue, Ebola, HIV. The immunogen of these viruses are always on the surface and heavily glycosylated. So we may take the same approach to deal with the problem there. In terms of cancer, that's a totally different story because cancer is still part of human cells, cancer cell. So it's almost impossible to identify a non-cell protein on cancer. You can only see the overexpression or mutation of certain proteins, but it's almost impossible to find a foreign proteins on cancer cells. That's why the development of cancer vaccine is much more difficult there. But it's possible to identify unique sugars on cancer cell, like what you say about sialic acid. And actually, we did find that. I showed the enzymatic synthesis of global H and SSCA4. And both glycolipids are embryonic on embryonic stem cell first, and then they disappear once differentiation started. And when you have cancer, they come out again. So we try to understand this, and we found that there's a new glycolipid pathway, which usually is not active in normal cells until you have cancer. And the key enzyme is called beta-3 GALT5. It's a galactose transferase which galactosylate a glycolipid. And once it's galactosylated, it will trigger the synthesis of global H and SSCA4. And these two glycolipids we call global series glycolipid, and they are involved in cancer metastasis. For example, in lung cancer, we know EGFR is the receptor involved in the connection between signal transduction and intracellular phosphorylation, and also TKI resistant is also related to the signaling of SSCA4 I mentioned about. Yeah. So once the signaling pathway is established, you will see that the intracellular network of SSCA4 signaling and EGFR signaling overlap. They communicate with each other. So even you apply the pressure to EGFR signaling, the virus will use SSCA4 to survive. But in a normal cell, we don't have that. We don't have that signaling pathway, the SSCA4. Right. So I think for cancer vaccine, carbohydrate could be a chance because carbohydrate are small molecules, and if the assembly line goes wrong, you create different structure there. Right. It's very different, and you can use small molecules as an epitope to design a vaccine. Yeah.
Okay. Anyone else has questions? We could probably take a couple more. I have one more question, if no one else. You talked about using a pseudovirus. Could you tell us some more? Is that the safety of that I'm assuming it's less infectious than the actual virus, but what are safety precautions and all of that you do with the pseudo virus?
Yeah, the surface is the same, but the inside is different. So we make sure that the virus is not infectious. We only create a surface which has the S protein there. So you can do the binding and infection, but the virus, the pseudovirus will not replicate.
Okay.
Yeah, so it's a safe model for S2 laboratory to assay the binding and infection of the virus.
Okay. Okay. And if no one else, I have another one. The how soon do you think this technology would bear fruit in like commercial products? What is your
This, this technology has been patented and available for licensing.
Okay.
And in the case of influenza virus, it's in the process, it's in development. But for SARS-CoV-19, it's available for licensing.
Oh, okay. Okay. Very good. Well, we want to thank you again. I've learned a lot of carbohydrate chemistry here today. And thank you.
Try not to talk too much about chemistry. So.
That's okay. That's okay. We've learned a lot from you. And we thank you and we wish you continued success, because your success is going to make this world a better place in terms of our fight against viruses and other diseases. So thank you for all of your work and your research.
Thank you very much. And thank you for listening. Yeah.
Okay. All right.